All right, as we take our seats to get going, the sound is on, Paul. It's just drowned out by the speaking. I have an announcement. This is concerning... Oh, thank you, Rachel. Good afternoon, everyone. I hope you had a good lunch. I'll hand over to my co-chair shortly to lead the next session, but I just have one important housekeeping, and it's to do with our field trip excursion tomorrow. Anybody who has expressed an interest in attending and hasn't registered, could you please see Hassini? Hassini, could you just raise your hand again? At your earliest convenience in the next break, I think will be fine. We just really need to lock in numbers. It is a juggle to try and make sure we've got enough room on the bus and that we don't have to tie Paul Johnson to the roof on the way down to Portsea. Okay, I think that's it. So see Hassini if you would like to go on the field trip tomorrow but didn't register. With that, I will hand over to Professor Simmonds to lead off the next session. Thank you.
Thanks, Tim. Right, let's try that again. Paul Johnson, everyone. So Paul Johnson is an infectious disease physician who for 30 years has led and co-led Buruli ulcer research teams in Victoria. His extensive clinical experience and developed the first Australian Buruli consensus treatment guidelines in 2007, which have since been updated with Dan O'Brien in 2014. He's a member of the WHO Technical Advisory Group on BU since 1998 and a foundation director of the Northeastern Public Health Unit, and it's immediate past director. Is the immediate past director of the WHO Collaborating Centre in Melbourne. We're looking forward to hearing your talk, Paul. The floor is yours.
Thank you very much, Rachel, for all those introductions. Okay, so I've got 20 minutes to summarise 30 years of research. Let's see how I go. So just to remind you that we have an exponential outbreak of Buruli ulcer in Victoria, and this curve, this is data that John Heyman had, goes all the way back to the 1930s. It's just N cases by year. The whole of the Mornington Peninsula is endemic, the Bellarine Peninsula, Chinjalong, it's in Melbourne. I'm going to mention in this talk Phillip Island, which is here, and Point Lonsdale, which is here. And the dinner is down here at St Andrews. So two important people you already know, Françoise Portels and John Heyman. This is a field trip photo taken during IMUC-II at Phillip Island, and you can just see IMUC-II written on John's badge, which proves this is not a fake photo. Now, at the time just before IMUC-II, which was 1998, we were investigating this big outbreak of Buruli ulcer in the eastern part of Cowes on Phillip Island. This outbreak had been recognised by the local family doctor there, Paul Flood. And you can see how it's all clustered around this golf course, which uses irrigation, spray irrigation, which was done with groundwater mixed with recycled water. And John Heyman had this very strong, clearly stated theory that aerosols transmitted M. ulcerans, wind action across contaminated wetlands. And this fitted really well with that model. Now, at the time, I entered the scene with a research group I was working in at the Royal Children's Hospital, and we developed IS-2404 PCR, originally to look directly in the environment, but it turned out to be quite a good diagnostic test, as a lot of you already know. Now, at the time we started testing the environment, I was really excited, and I thought if I could get a positive result from this environment, so we were testing the water system and the water downstream and from it, that would prove the hypothesis. And we did get some positive results, but now with age and experience, I realised that you can't actually ever prove a hypothesis at all in science. When you get a result which supports the model you already have, that's good, but what you need to do to be a good scientist is set up a model based on the evidence you see in such a way that subsequent evidence could refute that. Now, shortly after IMUC-II, this letter appeared in The Lancet from Françoise Portel's group, and she had taken IS-2404 PCR back with her to the Benin and Ghana endemic areas and looked in the environment there. Now, she opens her letter with Buruli ulcer caused by Mycobacterium ulcerans. usually occurs in the vicinity of tropical wetlands. Epidemiological studies have not obtained and not established a natural reservoir and mode of transmission. They collected 100 samples, water, weed, etc. And of those, the only 5 that were positive were insects. Now, meanwhile, at Phillip Island, the outbreak seemed to abate after we made some changes to the recycled water. People thought maybe that was the reason. But we started seeing cases on the other side of the bay. And this is an early case, a child of 18 months, whose mother was convinced that he had been bitten on the ear a few months earlier. Now, 10 years after cows on Phillip Island, we're at Point Lonsdale now, which is a similar sized town, and all these black dots are the human cases. And you can see there is a golf course, but it's not in the middle, and it didn't use recycled water. So you could say that a natural experiment in epidemiology had disproved the previous hypothesis that we had. So we were looking for something new. So we combined our clinical observations with that letter from Françoise Portel's and trapped mosquitoes. So in summary, we got, of 12,000 mosquitoes, about 4.1 per thousand were PCR positive. And we were just delighted. We thought, that's it. Everyone's going to believe us. But it took us a long time to get this established. But we just went on and started looking in the environment for where might the mosquitoes be getting the M. ulcerans. We PCR'd everything we could find, and one day we picked up this, which is possum excreta. The DNA level per unit in this was about 10,000 to 100,000 times more than we had ever got from any environmental sample before. Now, we then did this possum excreta survey, so that means you walk around and pick up possum excreta from Point Lonsdale and Queenscliff. And there were cases at Point Lonsdale, but not at Queenscliff. And you can see that the PCR positive possum excreta predicted or was associated with the human cases. We then trapped 42 possums at Point Lonsdale and were astonished to find that a quarter of them actually had Buruli ulcer already, and about half of them were excreting it in high concentration in their faeces. And these are some of the pictures of some of those possums. Now, bringing all this together, we published this paper in 2010. Janet Fife was the first author. And we stated a new clear hypothesis, and that is that in Victoria anyway, Mycobacterium ulcerans is not in swamps, but it is in the small animal reservoir of possums. They live in the trees, they bomb the environment with huge quantities of DNA in their faeces, probably living bacteria as well. And then mosquitoes can pick it up from the environment, or they can pick it up directly from possums. And humans can pick it up directly from the environment, or they can pick it up directly from possums. Now, that model turned out to be extremely robust because it has been all the subsequent research, and there's been two research groups working on this, all support the model. So it's standing the test of time, and we pretty much all agree on it now. Now, it's not that hard to get direct contact with the environment. So I just got a sequence out of thing. Yeah, yes. So what we didn't state when we put that hypothesis forward is how much M. ulcerans was transmitted from direct contact and how much was transmitted by mosquitoes. And this is very important because it determines the public health response that you take. Now, it's not that hard to get direct contact with Mycobacterium ulcerans in the environment, so let's imagine what would happen if most of it's transmitted by direct contact. Where would we expect to see the lesions? And here's just a possum pellet on the ground. Up to half of this is positive in endemic areas, and there's a human who's put their foot on it. Now, we get lesions all over the body, but we can also quantify that. So this is a human body map with, this publication was in 2017. Dr. Arvin Yaramilli is the first author. And you can, what he's done here is worked with all the doctors who treat Buruli lesions and mapped 649 of them onto a human body. The first thing to say is that only one of them was on the sole of the foot, and only 2 of them are on the palm of the hand. If you look at the back, this is the backs of the legs and the backs of the arms, and here's a blow-up of one of the legs. There's a lot of ulcerans lesions here, and this is not the part of the body that comes into direct contact with the environment. If you look at the front, there's a lot around the ankles, but there's also some on the legs and the arms. And we could argue about this, but I think we'd all agree that ankles is a place that mosquitoes like to bite. So what if we flip it and we say, what about if it's predominantly mosquitoes? What would we expect to see? Now, one of the early criticisms of our mosquito research was that all we had was a PCR signal. So we couldn't, people would say, well, how do you know it's M. ulcerans? And then they would say, okay, you know it's M. ulcerans, but how do you know it's the right strain, the same one that humans get? Now, these two questions and others have been answered very clearly in this paper in Nature Microbiology earlier in the year. And basically, this is one of the figures, and along here are Mycobacterium ulcerans genomes compared from different sources. And you can see overall that there are 4 strains at least in Victoria at the moment. And some of these points are human-derived M. ulcerans isolates. Others are reconstructed from mosquitoes. Some are from possums, some are from possum excreta. And the main point is they're indistinguishable. So what is in the human is the same as what's in the possum, is the same as what's in the mosquito. So how else could we look at this problem? Well, Victoria is one of the only temperate places apart from Japan where there's significant transmission of M. ulcerans. So we could take advantage of our clear mosquito season to investigate whether mosquitoes might be having an effect at the public health level. Now, our mosquito season lasts from November through to April. So if we look at notification data, we should be able to see a seasonal effect. Now, this is notification data. This is cases of Buruli ulcer notified by month and year for 6 years. There's almost 1,800 cases here. And in Victoria, it's a legally notifiable condition. So if you're the doctor, you must notify. And if you're the lab, you must notify within 5 days. So this is robust data. And it shows a cyclical pattern that's very prominent year after year after year. And the magnitude of this is very significant. It's up to 20-fold peak to trough difference. And any theory of transmission needs to explain this. However, when you look more closely, you realise what you get is the opposite of what you're expecting. So mosquitoes are most active in summer and autumn, and here you see that the nadir of cases, the lowest points, are actually in summer and autumn. So you could say on first principles that this interpretation refutes or disproves the hypothesis that mosquitoes are playing a significant role, unless you take into account the long known incubation period. And this has been measured twice. We've seen this graph before, but this is basically what happens when people from the health department interview cases who have brief contact with an endemic area. The median and the mean is around 5 months, but there is a wide range as well. I'm just going to skip over that one. Now, what we've done in this graph, this is Andrew Butchin's work. We have got the same curve that I showed you on the graph before. So in orange is Mycobacterium ulcerans notification data, but it's been log transformed and smoothed so that you can better see the cyclic nature of it. We've overlaid over the top of it in blue alpha virus notifications. Now these are Ross River virus and Barmah forest virus. These are known vector-transmitted mosquitoes. And they've also been, vector-transmitted viruses transmitted by mosquitoes. And you can see there is a curve there as well, a seasonal effect, but it runs counter to the Buruli curve. If you then ask the computer, what do you have to do to get these curves to align? Now, why would you do that? Well, in electronics and signal processing, that can be a problem that needs to be solved. And the answer turns out to be you shift the Buruli curve by 5 months, which is an extraordinary result because that's being generated by a machine analysing 2 curves, and it matches exactly what we found by shoe leather epidemiology about the incubation period of Buruli ulcer. So in conclusion, I think in Victoria, the reservoir and vector of Buruli ulcer is no longer a mystery, and it hasn't really been since 2010. At a public health level, Buruli ulcer is predominantly transmitted by mosquitoes from a possum reservoir, and this explains the location of most lesions on the body map, and it explains the strong seasonal influence on transmission. And it's where we should focus our public health efforts on mosquito season and mosquito bite prevention until we find a way of controlling it or getting rid of it out of our native possum population. And I'd just like to thank the very many people who have been involved in this research, in particular Professor Tim Stinear for his leadership of the Beating Buruli group in the last 5 years. Thank you very much.
Thanks, Paul. I'm sure there'll be questions.
I had a lot of questions from the reviewers when I submitted this.
Everyone's convinced.
I know some of you are reviewers.
If there are no questions, then...
Can I just add one more thing? I just wanted to show this slide which got out of sequence, but this is work from Koen van der Lanoot. It comes from a recent publication, and it's an incredibly detailed possum excreta map from Geelong. And what's been done here is if it's a black dot, they looked for a sample and couldn't find one. If it's an open circle, they looked for a sample and found it, and it was PCR negative. But where the colours are, they looked for a sample, found it, and it was PCR positive, and then occurs in these little clusters. And here in blue are the human cases. So there's this tight association between possum excreta that's positive and human Buruli ulcer. And finally, in this particular example, the samples were collected before the human cases were notified. So not only does it give you association, but it gives you direction and it gives you causation. Thanks very much.
Oh,
Paul, thank you for that, Paul. Great review and discussion. So just following on from this last piece of information, we found that there was about a 12-month lag between the possum sample environment being positive and then human cases. Do you want to comment on that?
Well,
We found on average.
the study that we did doesn't really have the power to show that. It was like we were surveying the possums, and then it just happened that human cases were notified shortly after.
well, the questions are coming now, Paul. Caroline?
So you find ulcerans in the feces of the possum, so it's alive?
Yes, look, is it alive? Probably it is. It is, yes.
So have you, has it been shown whether ulcerans is also found in the feces of human patients?
We have
with
looked for
Buruli?
that.
Do we have
Tim,
data for that?
we
Can
do-
it survive in the digestive tract? Do we
so
know?
with humans, as soon as we got the possum excreta, we went back and worked with Richard Phillips and others in Ghana and looked at 50 humans who had Buruli ulcer, checked their faeces, it was negative. So it's not the same thing. Can it survive in the gut? I don't think we can really answer that question with data, but I think it is alive. Or we've got some evidence. I think there's a RT-PCR that the Bowen people have, which suggests that it's active, but it's not culturable or very difficult to culture, and there may be a reason for that. It has been cultured once from the gut of a possum, but not actually from the fecal pellet.
Infectious disease physician from Bowen Health, and amazing talk and great work, Paul. Should be proud of all the work you've done. So it's always intrigued me the fact that the Phillip Island epidemic went away, but also that you had a theory and you made an intervention to address that, and it coincided with it going away. So have you got thoughts on what happened or how you explain that? Because it seems like you sort of worked it out and you...
Yes,
and
I
you
think,
actually...
look, what I think is that we stated a bold, clear theory that fitted the evidence we had very well. Then when we got another chance to see exactly the same thing, it didn't fit. So we then said events and data have disproved the previous theory, and we've moved on. But we don't technically know, like you can't prove that we were wrong. It's just that we've moved on, and I think we've found a much more fruitful field to what's the expression? A better field to mow than the one we had before. But it's possible that people were getting infected by aerosol. We haven't proved that we haven't completely disproved it, but looking back, I think that what happened at Phillip Island was just the same at Point Lonsdale. There were possums there that we found later. There were plenty of mosquitoes there that we never tested. And I think it was just the same story, but we fitted the wrong model to it. But the point is, if you don't fit the model, if you don't state clearly what's going on, then the next group can't come along and throw stones at it. And if you don't do that, it's not science. So that's the way I would explain that.
I think we had time just one more.
Thank you, Paul, for the fascinating presentation. My name is Michael Mleme. I'm an epidemiologist at Bowen Southwest Public Health Unit. My question is around your thoughts about incorporating Buruli ulcer surveillance in the routine ongoing mosquito surveillance we have in Victoria. Do you think that incorporating the testing for Mycobacterium ulcerans in mosquito surveillance would assist in predicting new areas of transmission?
I think it would, but I think it's simpler if you want to predict just to collect possum excreta samples, because that's such a reliable and stable and much easier to catch than mosquitoes. But if you're catching mosquitoes anyway for Ross River virus and other viruses, I think it is reasonable to add it in, depending on the cost. But it's important to point out that Buruli ulcer is being transmitted around Melbourne, around the peninsulas, where these other viruses, the alpha viruses, are uncommonly transmitted. And it's rare also to get the encephalitis viruses. They're up by the Great Rivers. So it's kind of like two different disease groups, and I'm not sure that it would be useful, but if you're wanting surveillance, I'd go for the possum excreta on the ground first. Thank you.
Thanks, Paul. So our next speaker is Lydia Mosi from the University of Ghana, and my sister from another mother, who has been a good friend over many years. She's in the Department of Biochemistry, Cell and Molecular Biology at the University of Ghana. Her lab exploits multidisciplinary approaches in silico genomics and functional lab studies to investigate gaps in transmission for BU. The floor is yours, Lydia. I'm looking forward to hearing your talk.
Good afternoon to everyone, and thank you for the opportunity to share some of the work we've been doing in Ghana and around West Africa to understand transmission. Okay, so I don't have my data slides, no problem. So that's my title, but I did have a slide which showed a picture of one of my mentors who really got me into this field when I was an undergraduate student. He supervised or co-supervised my undergraduate work, which was on transmission of Buruli ulcer, Professor Michael David Wilson, who sadly passed away a couple of months, maybe a month ago. He's going to be buried this weekend, so I had a picture of him to pay homage to him. So yeah, that was the picture. Anyway, so geographically in West Africa, when you take a look at work that has been done over the years, Mycobacterium ulcerans or Buruli ulcer has been found in quite a few countries. And the hotspots include Ghana, Cote d'Ivoire, Benin, and now more recently, more cases are popping up in Nigeria. If you take a look at the trajectory of cases that have been reported, you can see, as has been reported this morning, that it looks like cases are going down. But Kingsley said something that was really telling. Research really drives where you find the cases. When there's money for active case surveillance, you come up with more cases. But this is true. In the beginning, 2013, you had more cases, but now it looks like it's going down, and Cote d'Ivoire is still quite a hot spot. The conundrum of transmission still remains, and I'm really glad that in Victoria, at least in Australia, the top half has been sort of figured out, where we can clearly show this path and a possum somewhere here, and that's really good. And it's taken years of meticulous, carefully planned research to prove that. The rest of the world is still working on this conundrum. But what has come up with a lot of the research that has been done is that the bacterium is somehow moving between reservoirs or amongst reservoirs. Amoeba has come up strongly, insects have come up strongly, and then there's also the human host and whether or not they shed this into the environment. Now, several efforts have been made to try and understand transmission in West Africa. And a typical endemic area would look like this. And it has a lot of traffic from children who are swimming or playing, or parents who are going to fetch water, or fishermen who are going to catch fish to earn a daily living. This is a typical environment where you get loads and loads of Mycobacterium ulcerans in the environment. And most researchers have either gone based on active or passive case surveillance to identify regions that are classified as endemic. And they've taken a variety of samples from detritus, from biofilm, from plant, or the material that are on the surface of plants, and water, and have looked in this to see if they can find a signature of the bacterium in there. And most of the time it's DNA evidence. Some work, not a lot though, has also been done to try and see if animals have the presence of the bacteria in them and whether they could be potential reservoirs or vectors. But I think what is crucial is the fact that in the future, as we keep doing this work, we should start doing this with a One Health approach, because it's a complex situation and you find the bacteria everywhere. I took this data from a very comprehensive review that was done out of Cameroon in Professor Sarah's lab. Sorry, I was going to mention her last name, Professor Yango's lab. And it's a really good picture of the different molecular markers and assays that have been used to detect the presence of the bacteria in the environment. Conventional PCR, real-time PCR, using either the enol reductase domain, ER, which I think we started using in Pam's lab, the KR domain, VNTR typing, IS-244 profiling, IS-2606 profiling. And as you can see, the most positives have come from real-time PCR of IS-404 typing. And the cases, which is in the orange, also tell you the number that have tested positive for PCR based on these typing methods. But I do want to point out some key challenges that we've had trying to look at transmission in West Africa in particular. We have several environmental factors that challenge standardized sampling across the different countries. We have varying human activities, which you can't quite pinpoint as to which one is the most outstanding risk factor. We have several socio-economic factors that affect access to healthcare and therefore reportage. So we can't even have beautiful models like the ones that were shown by Paul earlier on. And then we have the whole conundrum again of climate change and how it impacts or influences tracking the presence of the bacterium in the environment. And finally, you have the sociocultural angle, which really prevents people from coming up and saying, hey, I have a lesion which is not painful, but I don't know whether this is witchcraft or it is something I should be more concerned about. Now, having said all of this, I want to highlight with 4 different angles that my research group in collaboration with others have tried to use to understand transmission. So about 10 years ago, we got funding from Wellcome Trust to do a profile on possibility of zoonotic transmission. And this work was done in Ghana and Cote d'Ivoire because we have similar geographic and environmental conditions. And the main objective was really to molecularly track the bacterium using a One Health approach. So we looked at rodents that were trapped in the homes of people who lived within endemic areas. And then we also looked at the environment around these people. And it was a huge collaboration between several research groups and hospitals in Ghana and Cote d'Ivoire. So what do we find? Over 2 years of sampling, we found obviously positivity of cases in human and in some of the rodents. And we used 2 ways to try and determine the presence of the DNA. We use Mycobacterium 16S RNA sequencing, as well as IS-24 for PCR. And you can see the numbers there. But what was really intriguing for us was one rodent which turned out positive for Mycobacterium ulcerans DNA, as well as VNTR typing. And we could overlay all of this information with the circle call-outs being cases that, or positivity that was found in the environment, and the rectangles being cases that were found positive in the humans. And you can see where, again, the rodent, which is the red triangle, was also found. And it's a really good way to show where the bacterium is and how it associates closely with human, rodent, and environmental cases. So there were quite a few people who were trained about through this program, and this is one of the beautiful things about doing research in the sub-region, to build capacity in this kind of area, and some publications came up from it. But I think what I want to highlight is the support that has come from the different groups. I trained in Pam's lab, and she was really instrumental in getting this work done with Heather, who came to help with some of the analysis and the training. Another work that we've done is to try and uncover the amoeba story. So amoeba have come up strongly as potential reservoirs. So we asked a research question whether M. ulcerans could survive in Acanthamoeba polyphagia, and this was an infection study. It was done by a master's student in my group, and he was able to show that, yes, the bacterium could survive beautifully in amoeba, and we could also detect the presence of the bacteria in the amoeba when we used red fluorescently labeled Mycobacterium ulcerans. But more importantly, we could show that it produced an infection, and passage through an amoeba made the infection even more severe. And you could tell that with the lesion that developed and also the histopathology that resulted. We've also tried to understand the genomic diversity or genetic diversity of different MPMs that we find in the regions that we've been looking at. And most of this work have been done by two students, Elizabeth and Magdalene, who is currently in Heather's lab. And what we've shown using VNTR typing is the fact that there are many profiles which would signify the presence of Mycobacterium ulcerans and related MPMs in the environment, but much fewer produce an infection in humans when collected from the same area. So the graph on the right is for the environmental MPMs, and the graph on the left is for the human cases. And then we've also been using nanopore sequencing to try and sequence the entire genome from the human isolates that we've been able to recover. And this is still work which is going on in collaboration with Rachel's group. And again, capacity building, training people to identify cases, demystifying the disease, sometimes even buying motorcycles to help people go and get cases or reported cases from regions or areas where there's no access to cars in general. And then finally, I want to talk about the in silico work that we've also been doing. And this is work that was beautifully started by Tim many years ago, where the first sequence came out from Aggie, and they were able to identify pseudogenes. And we think that looking closely at these pseudogenes could also help us identify the niche of the bacteria and where it's localizing to over time. So this student, Edwin Che Balfour, developed a bioinformatic pipeline which he used to compare lineages of isolates that had been about 172 isolates over the years. And he looked at what processes in the bacterium that had been affected by these pseudogenes. And as Tim identified many years ago, a lot of them have been in metabolic pathways and cellular processes that have been affected. But all of this gives us a very beautiful model of the fact that the bacterium is hiding from the sun. It is in an area where it is relying more on the niche that it finds itself for most of its carbon sources and most of its nutrients. It's undergoing a lot more oxidative metabolism, and it is still making mycolactone or not. We are not too sure. So that's work that still has to be done. But the takeaways from all of this is M. ulcerans really everywhere you look? Is that really the case? And if that's the case, then it's a huge issue, because then transmission is still going to be very elusive. But I think the importance of One Health cannot be overlooked. And Tim mentioned that earlier on this morning, that more research should be done in the One Health approach to look at all the possibilities of where you can get the bacterium to help understand transmission. There's a need to standardize the methodologies that are used to study transmission. And I think what Dr. Yango is doing in Cameroon with BU LabNet and Skin NTD lab network is a great way to standardize protocols which are used in testing for the presence of the bacterium in the environment, in humans, and so on and so forth. I think it's also crucial that we need to go beyond the DNA. We need to find the signature of the bacterium that indicates it's alive. So probably using more approaches that look for the transcript of certain key genes which are required for metabolism. And then I can't overemphasize how we need everybody on board, including herbalists, including the community, everybody else who is in the community, to help us uncover transmission. And I think definitely that blocking transmission is really the key to zero BU. So with that, I really want to thank all the people and the money that has supported this work. And an award from WHO, the Mwela Malasela mentorship program, has also funded the attendance of one of my students for this meeting. So thank you for your attention, and I'm happy to take questions.
It sounds like someone on Zoom as well. Lydia, that was magnificent, and hats off to you for being able to conduct transmission studies in your region, because, yeah, it's not easy, and what you've done is remarkable. Even getting funding for Buruli in any sphere is remarkable, so kudos, really impressive. How do you think about what we've discovered here and the transferability of that to your context in Ghana? I know you touched on it in your slides, but perhaps you could just speak a little bit more. Do you think you have to move far away from possums and mosquitoes when you think about transmission in Ghana, or is it just a small shift in the mode of transmission mechanism? Any further reflections there?
Yes, and it's funny because I hear Prof Wilson speaking through my ear right now, because if it was mosquitoes, I think a lot more people would have BU in Ghana, because they are everywhere. And there's no discrimination between urban and rural with the mosquito prevalence. So I think we need to look in a completely different direction. I think so.
So then, to prompt you a little further, we have mosquitoes everywhere here, and I guarantee that some of our mozzies will probably nibble on you during your time here. But maybe, I think here it's transient contamination of the mosquitoes with the bacteria. So might that be possible as well in West Africa? So that, the epidemiology is explained because the mosquitoes are only picking up the bacteria, or at least in a viable infectious state, for a short period of time.
I think my first answer still stands, because I'm thinking about frequency of bites. I'm thinking about, we haven't quite looked so much in fecal matter of rodents for me to say that it's a good place for the mozzies to be picking them up. But maybe if we do, we might see something. But so far, it might be a far stretch. It might be a far stretch.
Thanks, Lydia. Just to pick up on what Tim said, we have possums and mosquitoes and humans living close together all the way around the eastern seaboard of Australia, all the way up Brisbane, Sydney, New South Wales coast, and we only have Buruli ulcer in specific small areas. So everything is set up, but you have to add the super source. and the super source is the bacteria. If you tip the bacteria into the system, then the possums get it, then the mosquitoes transmit it to the humans. But if you catch mosquitoes or sample possums 5 kilometres away, they're all negative. And so I think that is what we're seeing here. We also spent, I would say, a decade PCRing all over the place and always finding something that was positive, something that was negative, would develop a new theory, and then it was negative the next time we tested it, until we got that gold standard, that incredibly high signal. And now looking back... Yeah. No, it's a comment, sorry. Now, so I'll get to the question. So what I was going to say is that what I think now about our area is that all the samples that were positive were from this one sample breaking down and spreading around, and presumably that same thing could apply in Africa. Have you got comments about that? You just have to find it.
I do have a comment, and it's a very... Okay, I'm making an analogy, and then I'll go straight to it. So when COVID happened, I mean, there was a huge prediction that Africa would be hit a lot, and a lot of people will get wiped out. But the opposite happened. And the opposite happened because most Africans are primed with what makes COVID so bad, the immune storm that happens. And so it didn't make an effective story like everyone expected. I feel it's the same with the Mosis. They have so many other things going on for them in Africa. I mean, they have so many other viruses and malaria. I mean, even if it's transient, they're busy doing something else, which is probably not related to... It might not be the right answer, but that is my comment right now.
Beautiful answers of the meeting so far.
I think we're running a little bit late. Is there a question from somebody who isn't Australian? That's a no. In that case, I think we'll move on just so that we can keep on time. So thanks very much, Lydia.
Thank you.
So our next speaker is Kim Blasdell, I believe, if I am correct. Welcome, Kim. Thank you very much.
It's okay. Hopefully I can work out how to use this.
Hold on. So you should just move this forward, and you should be...
I am there, so if I just do this.
Yes.
Okay, thanks. And please tell me if you can't hear me, because microphones don't like me. So thanks very much for the opportunity to talk today. I'm bringing you back to Australia and possums, and in particular I'm going to talk to you about a ringtail possum M. ulcerans challenge model and why we wanted to do it. So hopefully, no, that's what I was meant to start on. So possums, thanks Paul, you've done most of my work for me. Hopefully you're convinced that possums are a reservoir for Mycobacterium ulcerans here in Australia. There's quite a lot of evidence. Obviously the possums get the disease, they get ulcers like we do, but they also seem to get systemic disease, and more on that later. It's also found, DNA for M. ulcerans is found in their faeces, and there was a question asked, has it been shown to be viable in their faeces? Yes, we have done an RNA-based PCR assay that has found evidence of RNA suggesting the bacteria is alive in the faeces as well, at least in some. There's also been some evidence of direct transmission between possums and humans. So there's a case report that, I don't even know who that got there. Oh, no, sorry, not great. Yep. We've got sort of at least one case report already of direct transmission between possum and humans, and there's also been another recent case. Speak to Dan if you would like more details. You'll hear later how sort of the presence of M. ulcerans DNA in possum faeces can be predictive of future human cases. And then sort of going back to the Beating Buruli project that's been mentioned a few times already, we also did sort of like a large-scale case control study here in Victoria, and we found that case participants were more likely to report possums on their property, more possums on their property, more possum poo on their property, and M. ulcerans positive ringtail possums on their property as well. They also seem to be involved in driving the presence of where M. ulcerans is in the environment, as we found that properties that contained M. ulcerans DNA were more likely to have possums present, and things that possums like here, such as overhead power lines that they use as nice little superhighways for moving around, and also certain native vegetation. So, as we said, sort of like we know that they can become naturally infected and they can develop quite severe disease. But most of the data that we have about this is kind of like a snapshot. Like, we do have a little bit of longitudinal data, but that's quite hard and resource intensive to collect. So most of that data is just like a single time point from a single animal. Quite often, these are animals that are severely infected already. So this doesn't really tell us exactly how disease is progressing in these animals or how these animals are actually getting the disease. And obviously, there might, sort of depending on how transmission is occurring between animals, you might get sort of different disease progression and things. So we're thinking, well, can we look into this more by actually developing an experimental model, M. ulcerans challenge model? So there was a couple of studies done, I think back in the 1950s, on brush-tail possums, where they basically took, I think it was sort of like a bit of an ulcer from someone and stuck it in a possum. So we have no idea what the challenge dose or anything was. The brush-tail possums did develop disease, so yep, that was good, but nothing has ever been done in ringtail possums. And I guess at the moment, it could be a sample bias, but we have stronger evidence that ringtail possums are more involved potentially than brush-tails. Discuss that with people later if you like. So we had a few challenges about how we were going to actually do this, because although there are zoo collections of ringtail possums, we don't routinely keep ringtail possums in a laboratory environment, and the laboratory environment where we were going to do these studies was actually the large animal facility based at the Australian Centre for Disease Preparedness, which is a CSIRO high containment site down in Geelong. This sort of lab, it's fully contained, you have to shower out, it's artificial lighting and all of this, so we had to kind of work out, well, how do we keep these possums happy and healthy in this environment, and also sort of train our staff on how to handle them, how to collect samples and things like that. So we started sort of trying to develop protocols for them. We also had to decide, you know, we've got to start somewhere, what challenge route should we go for? And we decided to go for intradermal, partly related to the mosquito hypothesis, also because we know it's very successful in mouse models. And we also had to decide on what challenge strains. So we wanted to kind of make it relatively realistic. So we decided to go on a strain that's from the local area, so on the Bellarine Peninsula, collected back in 2004. And we also, thanks to Stephen, used some of the culture that he'd produced using his novel method that actually allows more accurate counting of how many bacteria are actually in the challenge dose. The hardest thing, though, as I said, there's zoo collections. They're not so keen on us using their possums to do nasty things to them. So we had to work out where we were getting the animals from. So the decision was made that we were going to go and collect them from the wild. And for our animal ethics reasons, we didn't want to be transporting them too far. So we decided to collect them in the local region. We obviously had certain suitability criteria, and that was things like that they were adults, they didn't have dependent young, that they were healthy, that there was no obvious illness or any major injuries, and also that they were M. ulcerans negative. The fun thing was that Geelong, which is where we were based and where we were going to get the possums from, had been declared an endemic area. So we had to do a lot of pre-screening of the sites where we were collecting the possums from, and then when we actually trap the possums, we had to then screen them again to make sure that they definitely didn't have M. ulcerans. So it was quite an intensive process. So the actual aims of our study was to try and get an idea of how M. ulcerans disease develops in possums over time. How quickly does this occur, and does this vary much between animals? And then do we get shedding sort of like either sort of like in saliva or in feces? So our study design was went out and collected 6 M. ulcerans negative and healthy ringtail possums, and we allowed them to acclimatise in our large animal facility. It was actually for 3 to 4 weeks because we caught the possums a lot more easily than we expected to. We did our intradermal challenge, and we used a kind of medium dose of M. ulcerans, so about 500 colony-forming units. Post-challenge, we were then doing health checks on a weekly basis until clinical signs started to develop, and then we increased that to twice weekly. And then when the animals reached either scientific or humane endpoint, then we did necropsies to look for evidence of any systemic disease. So these are our results. What we found was that we got lesion development in all 6 of our animals, and this progressed to an ulcer in 5 out of the 6 animals that we had. The other one may have progressed to an ulcer, but we actually had to humanely kill it because it started losing weight. We got some variability in the time to ulcer development, sort of with an average of 62 days. And we found that it was relatively consistent with what we were seeing in naturally infected possums. So the histology picture we've got up the top there is basically kind of an area of inflammation at the challenge site, and it was sort of surrounded by macrophages and lymphocytes. I'm not a pathologist, please do not ask me pathology questions. And the little inset bit, you can't see it here, sorry, I should have done like a full-size screen. This is just sort of showing lots of acid-fast bacilli within this region. So we also use sort of molecular techniques to have a look to see where else it might be present in the possum, and also histology to look at that as well. As we say, we obviously found DNA in all of the ulcers, but we also found it in a range of other internal organs as well. And in all animals bar one, this seemed to involve some element of the digestive system. And these molecular findings were supported by histology findings in 4 of the 5 animals. We found hepatitis in all 4 of those animals, and also pneumonia in 1 of those 4 animals. The other thing that's of interest is that in 1 of those animals, which was actually the animal where we had a DNA detection in blood, we also found acid-fast bacilli in a blood vessel, and this was the only place we found acid-fast bacilli anywhere away from the challenge site. So this maybe has some relevance for the mosquito transmission. The other thing I'll briefly say is we didn't really find any evidence of shedding. A little bit in the oral swabs, but this was like within the first 2 weeks, so it could be reflective of the possums sort of cleaning their challenge site. And in 1 animal, it was the one that survived the longest post the development of clinical signs, at about 28 days after the development of clinical signs in the fecal pellets. So it may just be that we humanely killed all of our other animals before they got to that point, or it may be that they require a different transmission inoculation route in order to develop sort of shedding. So I hope from this, and also thanks to Paul's talk, we can sort of all be in agreement that possums are an important kind of reservoir for M. ulcerans in Australia. We know that they develop disease naturally, but we've also shown that we can produce replicable disease using an experimental, in a challenge experiment. And what we're hoping to do is use this particular challenge model to maybe look at what differs between different inoculation routes, how possums may transmit the disease between each other, but also to maybe validate novel intervention methods such as a oral bait vaccination scheme, which if you'd like to know more about that, come and talk to either me or Daniel O'Brien. So obviously this was a huge group effort, so a lot of thanks go to the animal team down at ACDP in particular, and also Richard Plug, who's our pathologist, and then the guys at Buruli Health and at University of Melbourne. And thank you for listening.
Wonderful, thanks Kim, very interesting. I have questions. Are there questions on the floor?
You know the answers already.
Really, where were the spleens on the necropsies? You didn't show spleens, you showed liver and heart.
We didn't collect spleens, sorry.
My question was, did you do any pain scoring on the possums to see if the lesions were painless in the same way they're reported in
It's a little bit difficult because almost all of the handling was done under anaesthetic, but I know that the animal staff were kind of like poking the ulcers, but while under anaesthetic to see if there was any kind of response, and they didn't get any response, but it's hard to do.
But they weren't showing any, I guess a pain score was probably part of the
There
welfare
wasn't like
check.
the grimace thing or anything like that, no. They seemed, we did infrared monitoring overnight, and they were just continuing as normal.
Yeah, sounds sort of typical, doesn't it? Any other questions? Ria?
Yeah, thank you for the talk. I just would like to ask, you mentioned that the identification of the blood, the bacilli in the blood vessel can be kind of the evidence for the mosquito transmission. I did not understand why that can be.
So because mosquitoes drink blood, so they would be obviously putting their proboscis into kind of like capillaries and things like that. So if you are actually having the bacteria in the blood vessels, then it's they could potentially suck the bacteria up at the same time as sucking up the blood. Does that make sense? It doesn't look like it.
Try to think about that, but yes. No, I think that it's important to see that the ulcers can be in other places, in other organs, and also that might have some implications that we can also be looking into like the biomarkers in the blood for detection. So I think that there are some implications on that, but I did not see kind of this connection between the mosquito bite and the blood vessels. But just a question and a comment.
Yeah,
thank you.
Maybe we can chat after.
I suppose it depends on the size of the blood vessel, doesn't it? And I think we're going to have to move on because we're running out of time. I'm really sorry. Okay, yeah, so unfortunately our next speaker, Dr. Emma Hobbs, is unwell, and so Kim is very kindly going to give Emma's...
Try.
So go for it, and you'll have questions on it.
Let's see what happens here. I looked through these slides very, very briefly earlier, and I should say I haven't had any involvement with this study other than going out once to help Emma collect possums. But her study is basically focused on natural infections in possums. So I have a feeling I can probably skip over some of this because we've done that. Sorry, Emma. But yes, we'll go into... So, yeah, I suppose briefly, like I didn't go into this at all, but the two possum species we're particularly interested in here are the common ringtail and the common brushtail possums. And there's certain differences between the two. Like, we've definitely found evidence of emulcerans DNA and disease in both of these species. But ringtails in particular, they have a lower body temperature, which might be helpful for the growth of emulcerans than what brushtail possums do. They also have a different diet. Don't really know what that could mean, but one thing that perhaps is interesting is that ringtail possums are, she calls it something different to what I do. They eat their own poo, and brushtail possums don't. So as you can imagine, like if they're shedding emulcerans in their poo and then they're eating it again, they're probably continually redosing themselves with this bacteria. And ringtail possums appear to be more susceptible to Buruli ulcer. Okay, so Emma's study involved collecting several... I might just go on to the whole slide because I seem to know that a bit better. Yeah. So Emma's study involved collecting possums from a few different sources. So I think there were quite a few possums that were handed in to various veterinary clinics or to Zoos Victoria, but she also did a small longitudinal component as well. And she had a total of 26 possums that she actually looked at. 20 of these were possums that were handed in and had to be humanely killed, and so she did necropsies on those. As you can see, the majority of those were ringtail possums, 18, versus 2 brushtails. And then the longitudinal sort of study, what she's called the trap and release, she had 7 animals, of which I believe one of them did actually have to be humanely killed and necropsied, but the rest of them she was just sort of like anaesthetizing them, collecting samples, and then releasing them, and also checking to see if they had any evidence of lesions or anything like that. And then on the samples themselves, she was doing IS-2404 PCR and also culture. So the distribution of lesions on the possums are shown here. As you can see, they seem to be, a lot of them on the cooler regions, so sort of like on the limbs and on the tails of the possums. Here's some more lovely images. We all love our lovely images here, I think. So, sorry, this is going to be a very quick one. So what she did find was that in some of the more severely diseased animals, that the lesions actually went really, really deep and actually ended up exposing sort of tendons and or bones. So even if like the mycolactone is sort of preventing pain to some extent, you can still imagine if that's on limbs or it's on the tails and the ringtails, they actually use their tails as like a fifth limb, then that's probably going to be impacting their mobility, their ability to escape predators and things like that. And as we say, we just don't know if that's causing pain in these animals or not. Yes, and sorry, I did the other way around. So this is just a brief overview of what organs and things she found positive. And this sort of varied a little bit between some animals, but you can see in a few of the animals, these are the ones which are completely in red, that in the more severely impacted animals, they were actually positive throughout pretty much all of the organs tested, which suggests sort of widespread systemic disease. So as she said, not just skin deep. What she didn't find was any AFB anywhere. Where's the observed in stain? Oh, in any of the sections that she took for histopathology. But she also found some evidence of pathology in some of the organs, such as the liver and the lung, like as I've just spoken about in the experimental study. Obviously, this was a biased selection of samples because there are a lot of animals that were handed in, but you can sort of see that there seemed to be evidence of shedding in some. So she did sort of both oral swabs and cloacal swabs, and then also pouch swabs on the females. And you can, I think, sorry, the positive proportions are in red. So you can see there's a lot of animals that seem to have positive oral swabs and a reasonable number of cloacal swabs. But what's also interesting is that more than half of the females tested that she was actually getting pouch positives as well. And this maybe has some implications for, I say vertical transmission, that's not quite right, but sort of like dam to joey transmission as well. This is sort of the distribution of where she was getting her samples from, and the colours sort of indicate whether they were considered to have clinical signs, whether they were infected or uninfected. Sorry, I'm not quite sure what all of this is. So I think for these, the ones sort of highlighted in a purple circle, this was the first time that we had evidence of positive possums from these suburbs. And yeah, of the animals she tested, around 75% of them were actually positive for M. ulcerans. That's the breakdown. Sorry, I'm not doing a good job at this. So I think what we can sort of see from Emma's work is that it does look like M. ulcerans is very, very widespread in possums in the endemic area. From what she's found, it looks like it's potentially transmitted from multiple ways. Like we've obviously got the mosquito hypothesis. We've probably got like maybe direct transmission, sort of like bites and scratches. Possums are pretty territorial, so that could be a route. They could be, as I say, redosing themselves by eating their poo. Don't know if they eat each other's poo, no idea. I'm not sure either. I know certainly with koalas, mothers feed joeys their poo, so that could be a potential route if possums do that. And then with the positive pouch samples, then it's suggested that, yeah, mothers could be infecting their joeys as well. And because of the severity of the disease, this really is quite a concern for animal welfare in this area, but it's still not fully understood. And then I think I've briefly mentioned the oral bait vaccination that I've talked about before. And I think I might go, yes, if you would like to know more of the please check out Emma's papers, because that will probably be a bit more accurate than what I just tried to do. Thanks. I don't think I'll know the answers.
Okay, so thank you so much, Kim. We think you did a fantastic job. It's always difficult to present someone else's slides, so thanks for stepping in at the last minute. Our next speaker is Véronique Paris, who's going to present her work titled Understanding and Controlling the Vector in Victoria.
Yep,
You're already on the right slide.
Cool. Thank you very much. So yes, I'm Véronique, and I'm in the mosquito team at in the Beating Buruli in Victoria project that we heard so much of today. And Paul has set the scene for me nicely to say, while we're working out what's happening in possums and how we can control disease in possums, we should focus on mosquito bite prevention, and this is kind of where I'm doing my expertise here in the team. So I'm all about understanding vector species of diseases in general. And we are not just talking about mosquitoes in general, we're talking about one particular mosquito species here in Victoria. And I don't want to go into too much detail here because Stacey is going to tell you all about why we think this particular mosquito species is the culprit here, but a very brief profile so you know who I'm talking about. So Aedes notoscriptus, it's also called the Australian backyard mosquito because it breeds in our backyards. It feeds on humans and animals, so it's a very good candidate for spreading zoonosis diseases. It is invasive. It is within Australia. It is basically everywhere where mosquitoes can live, but it's also invasive to our neighbouring countries and more recently arrived in the US. It breeds in artificial containers. It's a mosquito species. We call them container breeder. It already transmits a couple of diseases that are important. So Ross River is an example, and it also is the major vector for hard worms in dogs. So it's also of veterinary importance. Its species status is quite complex, so we have some evidence that it's a cryptic species, and that makes sort of generalizations between populations of this mosquito species a bit difficult. There's also some evidence that we have a natural Wolbachia infection, the endosymbiont Wolbachia, in some populations of Aedes notoscriptus, but not in all of them. And we know from other diseases that Wolbachia can interfere with disease transmission, so that might further complicate the whole Aedes notoscriptus disease spreading part here. But what I'm doing is trying to understand this vector species, and it's important for two major sort of parts of this project. One is understanding why this particular mosquito species is implicated. That might be behavior, breeding sites, or all other sort of ecological features. And on the other hand, understanding these key ecological features to then go and control populations of this mosquito. And I want to show you a brief sort of overview of a more fundamental research project I've been doing to understand ecological features of this mosquito, and how this then led to a sort of research-based population control that we were doing in metropolitan Melbourne this summer. So first introduction is I was looking at dispersal and movement of this mosquito species, which is obviously quite important if we look at a pathogen vector, because it's quite important to know how far they could bring a pathogen. If I just breed a mosquito in my backyard, is it just hanging out with me and my neighbours, or if it's going across the shopping mall or even into a different suburb, that will definitely change where it might bring a pathogen. And it also impacts how we can control a mosquito population, if it's really localised or if we need to go a bit broader. So how we were doing this was using a genomics approach, because if you look at an organism that is small, it also flies. Sort of traditional mark release recapture studies can be quite hard to do. So we were doing a genomics approach here, and how we were doing this is we had the natural population of Aedes notoscriptus, and that's all down at the Mornington Peninsula, where we're all going to go tomorrow. And we offered them so-called oviposition traps, where they lay their eggs in. And then I collected all these eggs, reared them out in the lab, and sequenced their DNA. And then we were treating dispersal as an intergenerational process, where we exploit the fact that obviously parents genetically mark their offspring. So in the sequences that I got back, I looked for pairs of full siblings, half siblings, and cousins. And with that data, I was able to sort of look back into the past to work out what movement their parents needed to do to get them to the point where I collected them from. And out of time reasons, I cut a long story short for you today, and the results showed that Aedes notoscriptus moves about 10 times further than its cousin Aedes aegypti, the yellow fever mosquito, which is a lot better studied. And that was quite an important finding here, because when we look at those mosquito species that aren't really well studied, that are no disease vectors, it's often sort of generalized, sort of the ecological features we know about mosquitoes are sort of generalized from better studied species like Aedes aegypti, which is problematic if we see that they're actually behaving very differently. So these guys get around and they travel quite far, which makes interventions difficult. So there's just one identified sort of challenge in controlling this mosquito species, that they're high dispersal abilities. A second one that we found through breeding site surveys was that they're very good at exploiting what we call cryptic breeding sites. With cryptic, we mean everything that doesn't just like scream mosquito breeding sites to the average person. It might be bird baths, might be a saucer under a pot plant, might be overflowing gutters that I don't even know hold water because I'm not going up my ladder in the backyard every day. So that is challenging, and they're really good at using those. So normal source reduction programs might actually miss a lot of breeding sites that this species is really good at exploring. A third challenge here is that they go through up to 3 geotrophic cycles, which means they will blood feed up to 3 times, so the same individual is very likely to interact with a couple of different hosts. And it's also laying eggs up to 3 times, so they will contribute to the next generation quite a lot, just the one individual. So with these challenges identified, we were then trying to find a good research-based population control method that we were trialling this year in metropolitan Melbourne. And to try to overcome those, we used a novel approach. It's called the intercare station, which works as follows. It's basically a big sort of flower pot-sized bucket that attracts mosquitoes because it looks like a very nice, shady, watery breeding site. This water contains an insect-specific growth regulator, so every egg that's laid into this trap and then hatches within this trap won't emerge as a biting adult. So they're being taken out of the biting sort of generation. The second trick of this is why these females sit down to lay their eggs, they also pick up this growth regulator and then bring it to other breeding sites, these cryptic breeding sites that I mentioned before. So here we kind of put the mosquito to work themselves to find all these breeding sites that we might actually don't know exist, and reduce the number of biting adults coming out of those breeding sites as well. A third little trick here is that they also pick up spores of an insect-specific fungus, which cuts the lifespan of this mosquito in half. And that means that once those females are finished laying all their eggs and finding all these breeding sites for us, they will very likely not bite another human or animal, and they won't contribute to the next generation as much. All right, so that sounds all good on paper. Let's see if it actually works. So we were running this intervention in January, from January to April at metropolitan Melbourne areas as a randomised control trial. So, although it's a bit faded here, but this is basically next door to where we are right now. So we're looking at Essendon, Brunswick West sort of areas. Everyone who is from here probably lives in those areas. The very inner, very metropolitan areas. Everything in red here got treated with those stations, and everything in blue here served as a control to see if we have a difference between those zones after they've been treated. And those zones were chosen by all these possum surveys that we heard so much about. So we picked those areas because they had a lot of possum poo around, and we were expecting a lot of human cases arising. So we had a baseline before trapping, so the surveillance was all done on egg numbers. So we put all these ovi traps that mosquitoes are laying eggs in and counted the eggs 3 weeks before the intervention. Then we had those stations out for 8 weeks, and then we trapped afterwards in all of those zones to see if there are any differences. And apology in advance, this plot is very busy, but egg numbers are notoriously noisy to work with, but I'll walk you through it. So we're looking at the log of the average number of eggs per zone, and the thing that you really just need to focus on here is that all the treatment sites are in solid lines and all the control sites are in dashed lines. And we look at before intervention, we see that there's not a big difference between all of these controls and treatments, while we see after the intervention, in the first 2 weeks after, that all of those solid lines are below all of those dashed lines. So we did have less mosquito eggs in the zones that were treated with the intervention stations. I need to add here that we started with a very, very wet and warm summer, and we ended with a very, very dry and hot summer. So we had a natural decline of egg numbers throughout the project, but that's just sort of, field work for you here. It doesn't always just work in your favour. So that's a promising result on the entomology point of view. Obviously, everyone is now thinking of can we also see case numbers of humans declining in those areas, or is transmission delayed to when all intervention finished? And this is something we're working on right now. So as we all know, there's a big gap in actual transmission happening and them being reported. So that's a space to watch. But apart just from the disease point of view, throughout this whole project, I have noticed through talking to residents, because I did a lot of door knocking and adding those stations into people's backyards, that nuisance biting is something that is also very important for residents in their backyards. And I got a lot of people telling me they wouldn't use their backyards because of the mosquitoes they encounter in their backyards, which is quite sad, because having a backyard in the inner sort of suburbs is quite a luxury to have, and people should be able to use them. But with that, I'll leave you just to thank Ari Hoffman's group that I'm working in, and obviously everyone involved in the Beating Buruli in Victoria program. These are just the people I'm working with closely. Special thanks to Tim Stinear, of course, for letting me part of that project, and everyone else that I haven't mentioned are obviously very important parts of this team as well. Thank you.
Thank you, very interesting. Any questions from the floor? I can see one right at the back.
I can yell. You can hear me.
Yes.
Nyla from Melbourne Uni and Zoos Victoria. Just wondered, with the insect growth regulator that you were using, you mentioned it was insect specific. Is it mosquito specific?
It is dipterian specific, so it would affect flies as well, but in the way it's sort of applied in this particular sense, it's very mosquito specific, because those breeding sites are usually not shared with any other organism than mosquitoes, because those container breeders won't really breed in any sort of lakes or rivers or something. So it is, in the way it's applicated, is very mosquito specific, but overall the way it works is dipterian specific. Right?
Any more questions? No, great.
I ran away.
Thank you very much. Thanks, Véronique. So our next speaker is Stacey Lynch, who's going to continue to talk about Aedes notoscriptus. The floor is yours.
Fantastic, thank you very much. And I'm actually presenting on behalf of Peter Mee, who was the first author on a lot of the manuscript, first co-author of a lot of the manuscript that I'll be presenting, and he'll be here for the rest of the week. I thank our international colleagues for really showing me what the situation looks like in your countries. I'm going to be the first to present what it looks like in our country. So this is the Mornington Peninsula. It's beautiful, it's spectacular. You're going to see it tomorrow during the field work. But you were trying to pick a picture for a slide, so that was a good one. So a lot of this work is part of a really One Health team, and we were speaking at lunch about what is One Health. And to me, it's like bringing people, expertise from different disciplines together. People on the slide are just some of the people that were really involved in the mosquito work, but you can see the number of people on this manuscript and the breadth of the expertise that we had. to really support us try and untangle the mystery of how Buruli ulcer is, how Mycobacterium is spread throughout southeastern Australia to cause Buruli ulcer. Slides. So a mechanical transmission of Buruli ulcer, Mycobacterium ulcerans, wasn't new to this project. Led by Paul Johnson in, Paul, I can't see, it's too small what year it was, but really identified for the first time that Mycobacterium ulcerans can be detected on mosquitoes during an outbreak of Buruli ulcer. Tim Stinear then led some work with our colleagues in the US to really show, to set up a mechanical transmission model to show that we could actually cause a Buruli ulcer from injecting Buruli ulcer, Mycobacterium ulcerans into a murine, a mouse model. And we've all seen this hypothesis. And a hypothesis is always fantastic. It's a great, you build on scientific information, but then how do you prove a hypothesis? And so our challenge as the Breeding Buruli team was to see what scientific evidence we can do to prove this hypothesis. And we did stakeholder engagement, and we said, can we remove the possums? No. Can we remove the mosquitoes? No. People love the Mornington Peninsula, so they're always going to go there, so we can't remove the humans. So then we kind of thought, well, let's look back in the literature and find what else we can do. And there was a famous criteria established in 1960s, which looks, how do you look at incriminating a vector as epidemiologically important for a disease? And there's 4 main criteria that you need to meet. 1, that it acquires the bacteria from a source reservoir. 2, that you show the vector has close links with the infected host. 3, that you detect it more than once. You repeatedly detect this carriage of the pathogen on the vector. And 4, which is the experimental demonstration of transmission, which Tim and his team did before. So we sought out to try and address these different criteria of the hypotheses. So this involved a lot of surveying of mosquitoes, and so we collected over 75,000 mosquitoes over five years, identified 26 different species, and the predominant species when we identified them was Culex molestus and Aedes notoscriptus. It was fantastic to see Simon Couch there in the foyer, and Simon provided a small seed grant for a public health grant. So Simon's part of the Department of Health. He provided a small seed grant for us setting up the laboratory assay being transferred from Vidril and doing the initial surveillance in this area. And so, Simon, those 600 mosquito samples, they're in the table. And it also... so these mosquito samples, we didn't actually just look at the mosquito distribution and the species in the area. We also wanted to look at which ones are carrying Mycobacterium ulcerans. And so what I've shown here in the red slide is on the left-hand, the red box on the left-hand side, is there's 2 mosquito species out of that 76,000 mosquitoes over... almost close to 20,000 mosquito pools of between 1 and 15 insects were screened, and only 2 species came up. Aedes notoscriptus was detected every single year for that 5 years of sampling, with a total of 47 mosquito pools being positive. If you look at, on the table on the right is an indicator. So MLI is a way in which you can start quantifying mosquitoes per 1,000 mosquitoes, individuals, and with a confidence level. So as you can see here, between approximately 6 mosquitoes in every 1,000 mosquitoes in our surveillance sampling tested positive for Mycobacterium ulcerans. So just think about that. Like that's a huge number of an increase in frequency. Véronique is an absolute champion and has given you a really good rundown of Aedes notoscriptus. It's everywhere. It's in your backyard. It's really good at moving into new areas, and I saw it breeding in things from, I don't know, half the size of a drink bottle to a 50-gallon drum. So one of the questions that we haven't done is one of the questions that wasn't known is, okay, you've got a lot of mosquitoes, it's got a lot of Mycobacterium ulcerans on it, but what does it feed on? And so that was one of the questions we wanted to think about. and everyone in the room I saw has like probably slapped a mosquito and there's blood that's come out. So in that blood there's white blood cells, and those white blood cells we can actually look at the DNA and determine the species, and that's what we did for the blood-fed mosquitoes. They are pretty rare, so we identified out of that 75,000 mosquitoes there was 90 field-collected blood-fed mosquitoes in the area. Of that, I think it was 36, so we got a positive result. What we did was get the abdomen of the mosquito, squeeze it onto a piece of filter paper, use molecular techniques to try and understand what species it is. And this approach is really sensitive because you could find out whether or not it was a red wattlebird or a red dove. But interestingly, on 3 of these samples, we found mixed blood meals from humans and possums. And 2 of those were from Aedes notoscriptus, up the top here, and the third one is from Aedes rubithorax. And Paul, I'm going to preempt your question, which is like, why aren't we getting Mycobacterium ulcerans on Aedes rubithorax? And if you look at the 40% of the traps were Aedes notoscriptus, only like less than 1% of the traps were Aedes rubithorax. So it's kind of a take home as well. It's not only are they feeding on the host, but also are they at numbers that we could actually start seeing disease spill over, which is the human cases. Then there was some then we sort of enter in the fantastic molecular biologist, and I can't see Andrew anywhere, but he's chairing. Oh, you're there, oh, in front. He's chairing the next session, but Champion. So he developed a really sophisticated way in which we can start looking at, is the genome that we find on the mosquitoes the same as what you find in the humans and the same as what you find in the mosquito feces? Now, I'm sure a lot of you are in the field, in the room, aren't molecular biologists, but if you look on the table on the top, you'll see that the level of virus, so I'm a virologist, so I'm going to keep on saying that, the level of bacteria that we find in the feces varies about 1,000-fold between the mosquito samples and the possum feces. So there's 1,000 more fold bacterial DNA signals in the possum feces than there are in mosquitoes. So Andrew did his magic lab enrichment for the Mycobacterium ulcerans genome, and we could actually get enough genomic signatures from the different environmental samples to show that the bacteria is the same in the feces, on the mosquitoes, and in the human cases, which is a really compelling bit of information. We also realise that not only there are other things that bite you, and I suppose one of the hypotheses is it's just an insect that bites. And so working with the Barwon Health team, and great to see them here today, and Kim Blasdell, we're able to collect their bycatches during their case-controlled study, and we collected over 20,000 insects in this way, and this is mainly by people sending through the samples. Of these, we then went through and screened close to 3,000 biting insects that could pierce the skin, and of those we detected 2 flies that detected positive, and using molecular barcoding we could identify the species level of that fly, and when we speak with our veterinary colleagues and our entomology colleagues, it seems like this fly actually lays its larvae in decomposing flesh, and so we are hypothesising that it's the flies taking advantage of a dead possum, and possibly that's how the Mycobacterium ulcerans was detected in those. So we believe we've met the criteria. We've identified the bacteria from the source on repeated occasions. And I suppose so the impact of this, and it's a shame I don't know if the CHO is here, but this information and this project has really fed into the public health department on what surveillance strategies they're looking at, but what public health messaging they're sending out to the public that's evidence-based to try and reduce the number of Buruli ulcer cases in Australia. And Tim, I think that's me. Obviously, people on the screen are people that were heavily involved in the mosquito aspect, but as you can see from the manuscript, there's a magnitude of people involved in the work. So thank you very much, and happy to take any questions.
Lovely, thanks Stacey. Questions from the floor? Barbara?
Congratulations to the entire team. I was wondering, all these different Aedes, have you also looked at which other Aedes species may be vector competent for M. ulcerans, and does that shed any light on the African transmission cycle, or Aedes albopictus, which is coming north in Europe? Would that be a competent vector for M. ulcerans?
I'm a believer that it's because it's a mechanical transmission. I think there's a lot to do with if they bite at the same length, and their behaviour of how frequently they want to feed. Tim, you've got the microphone there, but in your study you did Aegypti, right? Yep. So it's been demonstrated for Aedes aegypti, which is another, obviously, a lot of, if you think about it, a successful vector is one that bites really a lot of things at really fast. So.
I could just follow up that we used Aegypti and notoscriptus, but only notoscriptus demonstrated competence for transmission. So, yeah, there's something going on there.
Yeah.
Could you speculate, I have a question, could you speculate then, Stacey, on why we see the discrepancy between, molestus dominant species, but only notoscriptus?
I think there's 3,000 mosquitoes in the world. We detected 30 of them. They all behave differently. Molestus is I don't think if a mosquito that will feed in a lot of different hosts is not going to fly over 10 different hosts to get to the one, but molestus is known to be more of an avian feeder. So you kind of wonder, is it flying higher? Is it is it flying lower? Is it just not interacting with those blood meals? They just want blood. They sort of don't feed.
Okay, one last question, Paul.
Stacey, you say it's mechanical, and I say that in my talks, but when I think about it, I don't know that it's mechanical. Do we know that it's mechanical, or could it still be biological? This is the vectoring of M. ulcerans by mosquitoes.
That's a good study if it hasn't been done already. Anything's possible, Paul. I suppose it's been demonstrated to be mechanically transmitted. Whether or not it be, maybe that's a question for, let's put that on your list, Mel, for the insectary. Yeah. Okay. So Mel's the team leader and the manager of the PC-III laboratory insectary, and so maybe it's on the list. There you go, Paul. Okay. We'll let you know.
I can see Lydia's desperate to ask a question, so we'll just squeeze one in quickly.
So, riding off the back of that, if it is indeed mechanical, then shouldn't there be a lot more of these vectors coming up positive or being the culprits?
That's more than a 2-second question. So in Australia, there are 2 to 3, 4 main vectors that drive all the outbreaks. They're the vectors that get to really high numbers, and they bite humans and they bite animals. And there's 4 of them. In the Morning Peninsula, it's only the breeding environment for one of them. In the Bellarine, the salt marshes are heaps, and the Gippsland, the salt marshes are heaps, and so, but there's only so many vectors that get to a high number and will bite different animals. Will bite humans, bite people, and I think that's the thing.
Thank you, Stacey. So I think it's a real take-home, isn't it, for us kind of human biologists, that mosquitoes are complicated, I think. That's the take-home. Brilliant. Thank you so much. Okay, the last speaker in the session is Eric Benbow from Michigan State University, and we're going to take an afternoon tea break after your talk, Eric, so no pressure, go quick.
Go quick.
I think if you just advance it, it should. If you just
this
press
doesn't work.
No, it does, but there you go. We'll talk you
Oh, okay. Well, thank you all for being here, keeping your attention to the tea break. Thank you, Tim, for hosting and organizing this, and everyone else who's been part of the local arrangements committee. It's great to see a lot of familiar faces. I haven't been to a WHO conference in probably 9 years, so it's really good. And congratulations on this amazing paper. The amount of work and the science that went into that is truly exceptional. I've been watching from the sides. I didn't review it. But I want to talk a little bit about some work that is a little different than what we've been hearing about. It's related to Mycolactone and mosquitoes, but it's also part of a larger project that we've been working on with a French group in French Guiana. And a lot of this work that I'm going to talk about with the data are from Joe Receiver, a PhD student that came out of my lab right around COVID. So some of it was truncated because of COVID and issues of getting into the lab and breeding mosquitoes in many ways. In that larger project that I'm talking about, this is where we switch gears a little bit. We needed to get money to continue to study Buruli ulcer. And one aspect is the EEID program, Ecology and Evolution of Infectious Diseases, from the National Science Foundation. But you have to ask very specific ecological or evolutionary questions, right? Without a lot of human potential impact. So what we did was come up with an idea based on a couple of different hypotheses that I'm going to talk about. And one is the novel weapon hypothesis. In this, we're going to focus on Mycobacterium ulcerans. A lot of it looked at the history of literature related to the evolution of Mycobacterium ulcerans, the acquisition of the plasmid, and the ability to produce Mycolactone. And without jumping into a lot of this, there's a big part in here of the ecological dynamics that may have led to some of the speciation events, right? From amphibians and frogs to humans. And so we wanted to ask those questions about perhaps Mycolactone had a different, or does have a different function in the environment outside of any kind of vertebrate host. And if you posit the novel weapon hypothesis, which was originally developed and tested, and it's controversial, for terrestrial invasive plants and how they spread to new environments. And basically, the introduced plants have these novel weapons. They're not called weapons, they just evolved these molecules or biomolecules to survive in their native habitat. But those molecules or biomolecules are really good at letting them establish in a new area because of naive ecological situations, right? So they can outcompete potential competitors. And it becomes this advantage to an invader. And so it's rampant. It's a theory in ecological community dynamics, invasive species biology. And so we framed Mycobacterium ulcerans within this context. Doing the same reading, though, of invasive species biology and pathology, which is weird to think about, you have both of those on your desktop, there was this other development of the coincidental virulence hypothesis for certain pathogens, that environmental pathogens, for instance, that they possess traits that coincidentally affect vertebrate hosts, right? And so the selection occurs outside of the host, perhaps in different niches, stable niches probably, providing the traits that allow them to exploit new niches, but also potentially persist, or maybe it's an advantage to disperse in vectors or reservoirs that haven't been identified yet. And so there's arguments that certain, I think Vibrio is an example, some Clostridium, that maybe they do have molecules that are being used in the environment, but when they get into a human host, those same molecules now cause pathogenesis. And so for Buruli ulcer, we thought, well, does this make sense for this system to maybe test these hypotheses or develop some experiments to test these hypotheses? And so if you look at neglected tropical diseases and you look at Buruli ulcer, probably a lot of underreporting, right? And for Buruli ulcer. But it's still probably not a huge number of people in the world. But if you start looking through the literature, especially in Africa and French Guiana, Mycobacterium ulcerans is found in a lot of different places where there aren't active cases. So you can go to non-endemic areas, no active cases, but you can still find Mycobacterium ulcerans in the environment, mostly in aquatic ecosystems, and that's where most of the sampling that I've seen in the literature and what we've done has taken place. And so we thought, okay, perhaps it's living in the environment in some way, and Mycolactone is a way that it does it. Well, COVID happened, right? We got funded for this 5-year project in 2019, and we were supposed to leave for French Guiana the day before international travel shut down. And so we were freaking out. We're like, we're going to lose our $2.5 million to study this. And so what do? Well, there was this obscure paper where it used IS-244 and found what they said was ulcerans along the southeast coast of the United States. So I said, well, I can get special permission from my university to travel to Mississippi with my colleagues Heather Jordan and Michael Sandell, maybe we should just go re-sample some of these areas and then use ER and some more specific markers to determine whether or not M. ulcerans is really there. Well, you can see from this map, out of our 9 sites, 5 of them have been turned positive. Heather's done the VT&R profiles on them. You can find them in multiple months along the southeastern coast of the United States. I only say that because we didn't really anticipate finding it there. We never even thought about sampling there. But once we did, and you look at the profiles, and if I believe the profiles are right, they start to match with more of the Mexican profiles as well for some of them. So it could be potentially out there in places in a really low level, right? We just don't know. There's lots of mosquitoes in Southeast US. We don't have any case reports of Buruli ulcer. So it's an enigma in many ways. And so that's what the background of this is, trying to understand mycolactone's function in natural ecosystems. And it looks kind of similar to this area. And so I'm actually here, one of the reasons I'm here, we want to start studying this in the Southeast US and use this model, right, as a skeleton for developing new experiments. We have a southern possum in the Southeast US that might be fruitful for new funding as well. So what do we know about mycolactone? We know it's immunosuppressive and cytotoxic in vertebrates. Mostly in vitro studies show that mycolactone regulation can depend on nutrient and resources. There's some interactions with fungal communities and fungal spore germination and attraction of certain fungi to mycolactone. There's been demonstrated effects on Staphylococcus aureus metabolism and gene regulation as well that came out of Heather's lab too. And so there seems to be a plausible hypothesis that mycolactone might affect other microorganisms in its natural environment. In the lab studies, not a huge number of mosquito lab studies looking at behavior, right? But in the Sanders paper, it was increasing mosquito laying behavior and host seeking without naturally occurring bacterial communities, like polymicrobial communities from natural water bodies, but just putting mycolactone into water and looking at behavior responses of insects. Variable effects on mosquito growth and survivorship, I believe that took larvae to adulthood. And then again, more variable effects of mosquito behavior with volatile organic compounds that were being emitted from solution with applications of mycolactone. So potentially, there is this indirect role, we think, hypothesized, that mycolactone is potentially using, or Mycobacterium ulcerans is using mycolactone to affect other microorganisms in, say, a complex biofilm that's full of algae, protozoans, other bacteria, and fungi, or in the rhizome of a plant, right, where there's a lot of other interacting organisms, or in the gut or the exoskeleton of an aquatic invertebrate, a snail, a bivalve, a biting hemipteran, potentially. But in that natural environment, very complex environment, we do know that mosquitoes are attracted to, and their behavior is dependent upon, odor cues, right? Whether that's finding a place to lay eggs or finding a host to feed upon. So the research questions that Joe wanted to, oh my goodness. Does mycolactone alter mosquito oviposition when introduced into a complex microbial community? Does it affect other aquatic microbes? And is there a temporal effect on mycolactone activity? And the first study looked at this in, I just skipped it. We did field oviposition studies in Benin, where we used a paired block design, took water from environmental sources, and used 2 concentrations of mycolactone, and then looked at the number of mosquito eggs that were in these container habitats that were placed in and around this University in Ouija. And what we found was a significant reduction, at least for that particular area, that time of year, in oviposition in the mycolactone-treated naturally occurring water. And there was differential changes on the relative abundance of the top most relative abundant taxa in the microbial communities using 16S and followed with ITS. And if you looked at the entire community structure You had very significantly different Mycolactone treated communities, bacterial communities, versus the controls as well, suggesting Mycolactone is likely having an effect on the entire community of bacteria in these systems. Followed up back in Michigan, he wanted to do this again, but we used oak leech leafate. So he basically brewed oak leaves, made a stew. A lot of people do this for oviposition studies for insects or mosquitoes there. And he incubated them at 4 different Mycolactone concentrations, and then used ITS and 16S to evaluate the bacteria in fungal communities. And then also looked at gene expression, but I won't talk much about that. Bottom line here, because I rambled way too much, treatment. So we made 4 different trials, different leachates produced different microbial communities, and we had a hard time seeing if there was a treatment effect of Mycolactone after 1 day. And you can see there was a significant effect. I don't really believe it after 1 day. And there's evidence to suggest some differential gene expression too. However, he ran it again over time, over 7 days. And this is beta diversity. This is the total community of bacteria, and the ITS showed the same thing. What you see is no difference in day 1. Day 3, the control and the lower concentrations start to separate out. Day 5, they really start to separate out. Day 7, they're very distinct communities, suggesting that Mycolactone is having some indirect or direct effect on multiple taxa in this complex microbial community. And so, with conclusions, the presence of Mycolactone, while a lot of this is preliminary, we need a lot of additional studies to determine whether or not Mycolactone is actually and functionally affecting other bacteria, and potentially how, what are the mechanisms by which that's happening. And so from there, I'll just thank my funding sources. Thank you all for your attention. I apologize for going over.
Thanks, Eric. Any questions from the floor? Everyone's desperate for tea. Tim, go ahead.
I could probably just ask one question. Are you using just for the people on Zoom, are you using physiological relevant concentrations of Mycolactone in your sort of micro environments you're trying to set up?
We don't know.
I second that question.
We based those concentrations off of some of the previous mosquitoes. I don't think anyone knows. We used the concentrations that were shown in previous studies to have an effect on some of the mosquito behavior, whether it was positive or negative, but it showed an effect. So that's the biggest question, is how much Mycolactone is too much, and then you just get a false positive, how much is not enough, and maybe we're getting false negatives after day 1 or 2, whatever.
Yeah, I mean, I think that's interesting, isn't it? Because how much Mycolactone is being released into the media of, even in a planktonic culture, compared to how much is still sticking around the bacteria. But really interesting. Any other questions, or is everyone desperate for a cup of tea/coffee? I sense people are desperate for sleep. Thanks, Eric.